The human body is made up of billions of cells. These cells form tissues that, in turn, combine to form organs. For all the organs in the human body to form and perform their functions properly, a number of processes need to run smoothly, using the instructions found in our genetic makeup. An individual's genetic material, known as the genome, is made up of DNA stored in the nuclei of all our cells. Let's try a few culinary metaphors to illustrate the composition and use of this genetic makeup. The human genome contains around 20 to 25,000 genes. They correspond to a collection of recipes that are used for the fabrication of the components that are needed for the proper functioning of our tissues. The genome is therefore like a huge cookbook, and the different tissues are the different restaurants, pizzerias, Chinese restaurants, etc., that are responsible for using the gene recipes from the genome cookbook to ensure that our bodies function properly. The genes are like recipes for making a dish, for example a pizza. The genes are composed of introns and exons. The totality of the genome's exons is like the list of all the ingredients needed to prepare the recipes in the cookbook. The combination of these ingredients is what makes it possible to prepare the many different types of pizzas. This collection of exons is called the exome. Introns don't provide any additional information, but they do contribute, from the same recipe, to the combination of different ingredients and thus to the making of several different pizzas. For example, the Regina pizza with tomato, cheese, oregano, ham and mushrooms, or the Margherita pizza with tomato, cheese and oregano. Different combinations that our tissues require to function properly. Although the exome accounts for only 1 to 2% of the genome, it is in the exome that we find the vast majority of anomalies known as variants or mutations, which are responsible for rare genetic diseases. A variation can, for example, make an exon disappear. Thus, an ingredient such as ham or mushrooms is no longer available, which means that the Regina pizza can no longer be made in our restaurants, the tissues that make up our organs. The remaining 98 to 99% of the genome, the introns and the regions that separate our genes, are called non-coding regions. These non-coding parts of the genome play an essential yet little-known role in regulating gene expression. In other words, the use or not of the different recipes. In fact, depending on the tissues, always equated to our restaurants, and depending on the body's developmental needs, lighter summer recipes or heartier winter recipes may or may not be used by the chef. Previously, this non-coding genome was considered to be of no interest and was referred to as junk DNA. But that's not the case. For many years, the tools used to identify the cause of rare diseases were not very effective. In the early 2010s, thanks to the development of new high-throughput sequencing technologies, a veritable revolution took place. Next-generation exome or genome sequencing makes it possible to find the cause of the disease, the responsible variant, in 50% of people with rare diseases. For the remaining 50%, carriers of a rare disease with no known diagnosis, how can this impasse be resolved? To return to our culinary metaphor, why is the recipe for a Regina pizza no longer used or served in a restaurant when that restaurant is Italian, and the Regina pizza appears on the menu as usual? This lack of diagnosis may be due to technical or knowledge limitations. Also known as variants of uncertain significance, or variants in non-coding parts, which are still poorly understood. They are still difficult to identify or to implicate in the disease. To take things a step further, the combined or sequential use of new techniques could help to identify a diagnosis. This is what we call omics, or even multi-omics. With this approach in mind, let's take a look at how we can move forward in the current state of knowledge.